Okay, I just wanted to go through this lecture and point out some of the big topics to try to help you understand these because I know there's a lot of words and there's a lot of details, but I'm going to try to simplify this as much as possible. So, we were talking about adaptive immunity. Remember, adaptive immunity is specific, and we also develop memory with our adaptive immune system. And we started talking about the process of humoral immunity, which is the antibody based immunity. And this is going to start when B cells will come in contact with antigens that are free or just extracellular in the solution in the body. Okay, B cells, as we know, have IgD and IgM on their surface, and when these surface antibodies bind that specific antigen, the B cell becomes activated. Now, activated B cells will undergo what's known as clonal expansion, which basically means that the B cell makes clones of itself. So you have an expansion of these clones. Um, and a subset of these clones will become plasma cells, which will produce antibodies against that specific antigen, or they will become memory cells that remember that specific antigen just in case those cells encounter the antigen again. Now, this whole process of clonal expansion usually requires the help of a T cell, specifically a helper T cell, or a CD4 positive T cell, which we'll talk about in a second. So here's just kind of the general process. We have our B cells that encounter their specific antigens. Some of those B cells will become activated and become memory cells, which will remember that specific antigen. And some of them will become activated and differentiate into plasma cells, which will produce antibodies against that antigen. Okay, so let's talk about how T cells help B cells proliferate and differentiate. So B cells are antigen presenting cells. Remember, the antigen presenting cells are B cells, dendritic cells, and macrophages. And these antigen presenting cells have MHC class 2 on their surfaces. Remember, those are the baskets. Now, when a B cell encounters its antigen that it's specific for, it's going to ingest that antigen and it's going to process it and then present it in the MHC class 2 basket. Okay? Now, helper T cells or CD4 positive T cells recognize MHC class 2. Okay? And they will dock on the B cell on that MHC class 2. And when they do that, they release these cytokines that activate the B cell, and that tells the B cell to undergo clonal expansion. All right? Now, this also allows class switching, which is when the B cell or the plasma cell starts to produce different types of antibodies. So remember that B cells, when they're not fully activated with the help of a T cell, they produce IgM only. But with the help of a T cell, they can class switch. They can produce different classes of antibodies. Okay. And this little note down here: B cells that are reactive against the body's tissues undergo clonal deletion because we don't want these B cells to be active against our own tissues. So this is a really good picture. This is showing that the B cell binds to its specific antigen. So this B cell is specific for this antigen. Okay. This antigen is brought into the cell where it's going to be processed. And then those processed components are going to be shuttled to the surface in the baskets, MHC class 2. Okay. And T cells are going to recognize that. And when they recognize that, they're going to dock on the B cell. They're going to release these cytokines. And that tells the B cell to undergo clonal expansion. Some of those B cells that undergo clonal expansion will become plasma cells, which produce antibodies against this antigen. And some of them will become memory cells, so they will remember that antigen. So we talked about the difference between T-dependent and T-independent antigens. The big takeaway: T-dependent antigens are antigens that require the help of a T cell in order to produce different types of antibodies. And these are typically proteins. T-independent antigens, they can stimulate B cells directly without help from a helper T cell. These are typically carbohydrates or lipids. The only problem here is the immune response is typically lower, and you don't have any memory B cells produced. Okay, then we talked about the actions of antibodies. So as we remember, antibodies will bind to specific portions of antigens known as epitopes or antigenic determinants. And once they do that, a number of things can happen. Agglutination, this is when antibodies cause antigens to clump or agglutinate together. IgM is very good at this because IgM, remember, is like a hand. Each finger can bind antigens. So when that happens, they can cause these antigens to clump and then kind of precipitate them out of solution. Now, opsonization. Antibodies can act as flags. So things like IgG antibodies can act as flags, which will, signal to phagocytes that, hey, come destroy me. Okay, and then the other two that I told you to know, activation of complement. So IgG or IgM antibodies can activate complement by the classical pathway. And then neutralization. So antibodies can bind to microbes or bacterial toxins and block their activity or block their ability to bind to cells and colonize cells. So those are kind of the big four actions of antibodies. Okay, so again, agglutination causes the clumping of these antigens, activation of complement, opsonization, so they can act as these flags, which signals these phagocytes to come consume the foreign antigen, and also neutralization. Okay, then we talked about cell-mediated immunity, which is kind of the other arm of adaptive immunity, and this is dealing with T cells. And of course, T cells, remember, they mature in the thymus. So we're going to talk about a couple of classes of T cells, starting with the helper T cells. Okay, these are also known as CD4 positive T cells, and we already know that they're very important in activating B cells to undergo clonal expansion and to produce different types of antibodies. So they also recognize antigens presented on MHC class II on macrophages as well as dendritic cells, which activates them. Remember, MHC class II is found on B cells, dendritic cells, and macrophages, the antigen-presenting cells. Helper T cells recognize MHC class II. Okay? And basically, the big picture here is that no matter what the antigen-presenting cell is, as long as it's presenting MHC class II, helper T cells will recognize that. When they recognize it, they will dock to that MHC class II. Okay. They will send cytokines that activate those antigen-presenting cells, and then they will in turn become activated themselves. Okay. So that's kind of the big picture here, and that's the level of detail you should be familiar with. Besides the whole thing with the B cells, because that's more specific. We talked about that specifically, but just in general as a big picture. Helper T cells recognize MHC class II, which is presented on antigen-presenting cells. When helper T cells bind to that MHC class II, they activate the antigen-presenting cell, and then they will become activated themselves. Okay. When they become activated, some of the T cells will become memory cells, and some of them will produce cytokines, which will have a range of other functions. It's more complex than that, but the big picture is all we really need right now. Okay. Then we talked about cytotoxic T cells, or CTLs, or CD8 positive T cells. They tend to recognize cells that have been infected with an intracellular pathogen, so things that are inside, so like viruses and some intracellular bacteria. And they recognize antigens presenting on MHC class I, which, remember, MHC class I is found for the most part on all nucleated cells in the body. Okay, so that's what these CD8 positive T cells will recognize. Now, these CTLs will attach to a target cell, thanks to MHC class I, and then they will release what's known as perforin. And perforin will put holes in the cell that they've docked on. Once those perforations are made, granzymes will enter that perforin and will basically trigger the death of the cell. Okay, and that's known as apoptosis, which is programmed cell death. So as this picture shows, here is a target cell. It could be any nucleated cell in the body. Let's just say that it's a respiratory cell and it's infected with a virus. Okay, so these antigens will be presented on MHC class I. That's recognized by the CD8 positive T cell. That CD8 positive T cell will bind to that MHC class I, will release perforin, which will make these pores, and then granzyme will enter inside those pores, and that will kill the cell. Okay, very important. They're also effective against some tumor cells. Alright, then we talked about the Tregs, the regulatory T cells, which is really a special type of helper T cell. But their primary job is to kind of fight other T cells that are kind of reactive against their own tissues, so they might be getting a little squirrely, and these regulatory T cells will step in and say, stop, you got to leave, our own tissues alone. They have other jobs too, but that's kind of a big picture topic for Tregs. Okay, as we know, adaptive immunity is characterized by memory. Okay, it's specific and it has memory. And that's different than the innate immune system. So we're going to talk about kind of this idea of memory. Now, when a B cell or a T cell are exposed to their antigen for the first time, what happens is a primary response. Okay? And this primary response is characterized by production of IgM, which makes sense because that's always the first one. And then you have a slow rise in IgG, and that's because you start to see class switching. All right? Now, in the secondary response, this is when these B and T cells encounter their antigen for a second time, or a third time, or fourth time, whatever it may be, after the first time. And this is characterized by a similar response of IgM because there are some B cells that will be experiencing this antigen for the first time, but a lot of the memory cells that were produced from the primary response are starting to recognize this antigen. When they do that, they produce very rapid amounts of IgG, which is shown by this picture, which is really good. So we have initial exposure to an antigen, then we have a rise in IgM first because it's the first one produced. Then we have a slow rise in IgG as class switching occurs. Now, on the second exposure to the antigen, we have a similar response in IgM because some new B cells will be experiencing that antigen for the first time. But those memory cells that were produced back here, when they recognize this antigen for the second time, they can just go ahead and produce IgG right away. And so you can see it's a much stronger, much more rapid response. All right, then we talked about the types of adaptive immunity. So we have four main types. We have naturally acquired active immunity. If you see that word naturally or naturally acquired, that means a human body is actually making antibodies. Okay. Now, if you see artificially, that means these are produced in the lab. Active means that the person's immune system is actively mounting the response, and if you see passive, that means they are just receiving the immunity. Okay, they don't have to do anything to produce it themselves. So naturally acquired active immunity, this is, your typical exposure to antigens. Okay, you get a cold and then you get over it. All right. Naturally acquired passive immunity, this is really when a mother is passing antibodies to a fetus or an infant through the placenta or breast milk. Artificially acquired active immunity, this is vaccination. Okay. It's artificially acquired because it's made in a lab. It's active because your own body mounts the response. And then artificially acquired passive immunity, this is the injection of antibodies that came from an animal or a human who is immune to a disease. Okay. And this chart just kind of breaks that down, just shows it in a different, more visual manner. Okay, then we talked about hypersensitivities. And hypersensitivity is basically when the response to a specific antigen known as an allergen is more than what would be expected if it was just a normal response. Okay, we talked all about the hygiene hypothesis. I got up on my soapbox and wore it out, and then we talked about the types of hypersensitivity. So let's just point out the big topics here. So type 1 hypersensitivity reactions. These are the anaphylactic reactions. These ones occur very quickly. And they tend to involve IgE. So those are kind of the associations you should make. Very quick, they're anaphylactic reactions, and they involve IgE. Basically, IgE antibodies are found on the surfaces of mast cells and basophils. When the antigen binds to that IgE, those mast cells and basophils will release their contents of their granules, which contains things like histamine, serotonin, and prostaglandins, leukotrienes, the list goes on. Anaphylaxis, when you see that term anaphylaxis, this is just basically a severe systemic type 1 hypersensitivity reaction. All right, and this is just showing the pathogenesis. You don't need to worry about it. Just make the associations. Very fast acting. IgE antibodies, things like that. All right, then we talked about the type 2, which are also known as the cytotoxic hypersensitivities. And basically here you have IgG and IgM antibodies that bind to a cell. And when they bind to a cell, they activate complement. Okay, this is going to be different than the type 3, as we'll see. So that's kind of the big takeaway here. These are the types of reactions you'll see if you have ABO blood group mismatches or Rh factor incompatibility. Okay, and then the type 3. So we're going to see how this is a bit different than type 2. So remember type 2, you have IgG and IgM antibodies that bind to cells and they activate complement. In type 3, which are also known as immune complex reactions, you have antibodies that bind to free or soluble antigens, not a cell. And this is typically just IgG. Now what happens is these antibodies will bind to these antigens and they make antibody-antigen complexes. And these complexes form more quickly than they can be cleared from the body. What happens is they accumulate in the tissues. And when they do that, they attract other immune cells and induce more inflammation. So you see this with autoimmune diseases. So things like lupus, things like certain types of glomerulonephritis, rheumatoid arthritis, all can be they fall under this umbrella of type 3 immune complex reactions. So as you can see here, this would be, in the glomerulus of the kidney, we have these antibody-antigen complexes that are kind of depositing themselves in the tissues in this endothelial layer, and then that attracts immune cells, which just makes the inflammation worse. So it can be pretty nasty. And then type 4. These are delayed type reactions. These are delayed cell-mediated reactions that involve T cells. So that's a big buzzword for type 4 reactions. These ones take a long time to develop. But basically, you have a sensitization step, which is when you first encounter this allergen. And these allergens are phagocytosed by macrophages and presented to T cells on MHC class 2. Now the reaction is delayed because on your next exposure to the antigen, these T cells will kind of activate cytotoxic T cells, which will release destructive cytokines. And this is what you see with poison ivy, TB skin tests, all of that sort of stuff. But the big takeaway, these are delayed type and mediated mostly by T cells. And we talked about this briefly with the allergic dermatitis that you see with poison ivy. All right, then we talked about autoimmunity really quickly. So an autoimmune disease or autoimmunity is just when our body recognizes our own tissues as foreign, and that's bad. The ability for our immune cells to recognize our own antigens and not react to them, that's self-tolerance. Self-tolerance is a normal state. We want our immune cells to recognize our own tissues and leave them alone. Okay? Now, when that self-tolerance breaks down, that's autoimmunity. That's the basis of autoimmunity. Most autoimmune diseases will affect women more so than men. That's not the case for all of them, but the majority of them. Why that is, it's not really known, but there is probably, hormonal components and other genetic components that play into that. Now, a lot of autoimmune diseases can be classified as type 2, 3, or 4 hypersensitivity reactions because that's how they kind of present. And we talked about some of those. So, for example, the type 2, the cytotoxic-mediated autoimmune diseases, these are things like Graves' disease and myasthenia gravis. Okay? You don't need to know any of the details about those diseases right now for this class. Just understand that they represent kind of these type 2 hypersensitivities. As far as type 3, those are the immune complex hypersensitivities, things like lupus and rheumatoid arthritis. And then the cell-mediated, the ones mediated by T cells, the type 4 hypersensitivities, things like type 1 diabetes, psoriasis, multiple sclerosis. Those are kind of characteristic type 4 hypersensitivities. The difference here between these and the other hypersensitivities is that the hypersensitivity is to our own tissues, and that's, of course, autoimmunity, and that's bad. So we talked about different types of grafts or transplants. So an autograft, this is when a tissue from your own body is used for another portion of the body, like a skin graft or like a bypass surgery for the heart. Isografts, this is taken from one person of the same genetic makeup, so an identical twin. So you got a graft from an identical twin. Allograft is a graft from another person, but they are not genetically identical. So this would be just like your typical graft or transplant from a donor. And then a xenograft, this is a graft from another species. So for example, if a human was receiving like a heart valve from a pig, that would be a xenograft. So graft versus host disease, like I told you in class, the first thing you should think when you hear graft versus host disease is bone marrow transplants. Because what happens here is somebody receives a transplant, a bone marrow transplant, from another person, and that bone marrow has competent B and T cells. B and T cells that are able to fight. So you're taking somebody else's B and T cells, putting them into your body, and of course those B and T cells are going to recognize every single one of your tissues as foreign, so they can just go haywire. And the problem here is the complications can be fatal. All right. We talked about ways to kind of get around this, but the big takeaway: bone marrow transplants, competent B and T cells, they fight the person that's receiving the transplant. So immunosuppression, these are just ways to decrease the body's immune response. So immunosuppressive drugs are used purposefully for autoimmune diseases or for transplantation, or even kind of allergic diseases like asthma. Can also be a side effect of treatment, age and disease. So obviously things like cancer treatment, as we age, we become more immunosuppressed. Diseases like cancer and AIDS are naturally immunosuppressive. And then some of these agents, glucocorticoids like prednisone, used for a wide range of conditions. The biologic drugs, so these are kind of the new kids on the block. They're used for autoimmune diseases. Those are things like Humira. Cytostatics like methotrexate, which is kind of like a low-dose chemotherapeutic agent. And there's many, many more. So we talked about cancer, which is really, cancer is characterized by uncontrolled cell growth and cells that have the ability to spread. Cancer at its base is really a genetic disease, but it's also at some level a failure of the immune system to recognize the cancer. And a lot of that is due to the cancer itself. Cancer develops very unique ways to evade the immune system. Normally, we have immune surveillance, which is done by cytotoxic T cells. They can recognize cancer cells and destroy them, okay, assuming that they can see them. If they can't see them, which happens in later stages of cancer, then they can't kill them. Or, sometimes the cancer just overwhelms the immune response. Okay, then we just talked about immunodeficiency, which is where there's an insufficient or absent immune response. So it can be congenital or acquired. So congenital immunodeficiency present at birth. So things like genetic diseases like DeGeorge syndrome or SCID, we talked about those briefly in class. Then, of course, acquired immunodeficiency. This is acquired through either drug treatment or through disease. So, many cancers are, you acquire immunodeficiency due to your disease state. AIDS, of course, is definitely an acquired immunodeficiency, or, the treatments that we talked about with immunosuppression. All of those are forms of acquired immunodeficiency. Okay? As always, if you have any questions, please let me know.